Hi, my name is Hedi Kari. I am currently a postdoctoral researcher affiliated with the CNRS and the University of Bordeaux, and today I will present my work on dynamic synthetic cells based on liquid-liquid phase separation. The research work I'm doing in my postdoc concerns the auto-assembly of de novo life, understanding certain biological processes via in vitro reconstruction using elemental building blocks, and elucidating physicochemical principles allowing an ensemble of inert molecules under specific conditions to auto-organize and produce an autonomous system. We are not only looking to reproduce static systems, but to create dynamic processes. Last interest concerns the development of novel intelligent colloidal systems for future applications in health and environment. So what are the necessary conditions for life to emerge? We can reduce their number to three. First, compartmentalization. It permits the localization of solutes in space and time and maintaining chemical gradients. Metabolism, the number of reactions required coupled to an energy source. And last, self-replication, the ability of a system to create copies of itself with a probability of presence of error, leading to Darwinian evolution. The goal is to develop new chemical systems that are compartmentalized, contain either one or both of the other elements, to mimic a living cells. To construct such systems, we are inspired by what happens in real cells, beyond membrane-bound compartments, including the cell itself and many other organelles which scientists were able to reproduce as vesicles. There is an emergent paradigm of intracellular compartmentalization in form of liquid-liquid droplets that don't possess a membrane, known as membraneless organelles, that are formed via liquid-liquid phase separation involving proteins, polynucleotides, and are present in many biochemical processes. Despite the absence of a physical membrane, these organelles can sequester or exclude biomolecules in a selective way, regulate enzymatic reactions. Their formation and dissolution is precisely controlled in space and time. Such particular properties encouraged us to investigate in depth these emergent systems. To construct such systems in vitro, we rely on liquid-liquid phase separation phenomena that can be either segregative with neutral compounds or associative phase separation, also known as complex coactivation, which relies on the association of charged species forming polyelectrolyte-rich phases in dilute aqueous phases. We mainly are interested in complex coactivation because it permits the construction of good models of membraneless organelles and protocells. My research in the group concerns mainly the development of responsive coactivates, including enzyme-regulated and light-actuated coactivates, and I'm going to talk about these two projects in this presentation. We first aim to develop a system where the formation and dissociation of coactivates is directly controlled by one of the scaffolds of such system. Coactivation between a polycation DA-dextran and an enzyme, glucose oxidase. Initially, we showed that these two components are able to form droplets at physiological pH, where the number of positive and negative charges are equal, but they can't form coactivates at high or low pH due to a charge mismatch. The interest in glucose oxidase is its ability to oxidize glucose into gluconic acid, inducing a pH decrease in the system. We showed that starting from high pH, GOX was able to control its own phase separation to form either stable or transient droplets, depending on the amount of glucose supplied. At low glucose concentrations, pH drops from 9.5 to physiological pH, inducing the formation of coactivates that are stable for a long period of time, as shown in this video acquired using bright field microscopy. Whereas, if we supply our system with a higher amount of glucose, we cross the existence domain of these coactivates, meaning they form, grow, then decay since the pH becomes lower than the physiological pH at which they form. We then decided to complexify our systems and for multiphase coactivates composed of this pH responsive phase and another non-pH responsive phase, ATP polylysine. At high pH or low pH, we only observe the existence of a single phase droplet, whereas at physiological pH, we noted the existence of a two-phase droplets as shown via confocal microscopy. Starting from high pH and depending on the amount of glucose supplied, we can form either stable multi-phase droplets or transient multi-phase droplets. And in this video, I'm showing the multi-phase stable droplets if we supply the system with a low amount of glucose. And here we can see the formation of the multi-phase droplets that remain stable in time. And if we supply our system with a high amount of glucose, we can see the formation of these multi-phase droplets that nucleate, grow, then decay. And at the end, we are left with the ATP polylysine phase only. So in this project, we clearly showed that in response to an enzymatic activity, we can control the formation and dissociation of coacervates, meaning the enzyme can regulate its own phase separation. In a second project, we are looking at developing light-stimulated coacervates to control spatiotemporal resolution. With UV light, we were able to dissolve the droplets, and under visible or blue light, we can reform them. So how does this work? The idea is to assemble these coacervates starting from photo-switchable molecules based on positively charged azobenzenes that interacts with double-stranded DNA. Under UV light, we induce the isomerization of trans-azo to cis-azo, which reduces its affinity to DNA, leading to coacervates dissolution. We then could reform these droplets upon shining blue light that drives back the photoisomerization from the cis form to the trans form, and therefore control in space and time the formation and dissolution of such system. As shown in this video, where we start with the initially formed droplets, and upon shining UV light, we can fully disassemble them. Then, if we shine blue light on this system, we can reform these droplets. Such technique offers a great spatial resolution where we can selectively disassemble the droplets, as shown in this video. We next sought to control the photo-induced trafficking of fluorescent oligonucleotides between two different populations of coacervates. We designed an experimental system in which oligonucleotide containing photoresponsive coacervates and payload-free non-photoresponsive droplets were physically separated by a semi-permeable barrier and then used UV light to induce transfer of the oligonucleotide between the two populations. Corresponding optical and fluorescence microscopy images showed the presence of discrete droplets in both populations, but only trans-azo DNA droplets displayed red fluorescence due to retention of the TAMRA ssDNA. After shining UV light, the dissolution of the photoresponsive droplets resulted in the release of the fluorescent molecules, slow diffusive transfer across the hydrogel, and graded recapture in the population of non-photoresponsive droplets. As a continuation of this work, we sought to develop other photoresponsive systems. Successfully, I was able to assemble photoresponsive coacervates based on either positively charged or negatively charged azobenzenes that I synthesized in the lab, and counterparts that includes ATP, polypeptides, synthetic polymers, and proteins. And in this presentation, I will show some of these results. Based on the same logic, we formed light-responsive coacervates from ATP and trans-azobenzenes that is positively charged and that can be fully disassembled with UV light. And can be reassembled upon shining blue light. on the system. Such coacervates are able to sequester and exclude different fluorescent molecules, and we could control the quantity of formed coacervates by controlling the percentage of trans-azo initially present in the system. An interesting observation is that we only were able to form these coacervates with ATP and not with ADP or AMP, which paves the way to create multi-responsive coacervates that can form and dissolve upon phosphorylation-dephosphorylation reactions of ATP and with UV and blue light. I was also able to form light-responsive coacervates with trans-azobenzene and glucose oxidase based on the same rationale, and I am working towards expanding this system to include other types of proteins, including intrinsically disordered proteins. Inspired by how the cell's nucleus is organized into multiphase condensates, we looked at the formation of multiphase light-responsive coacervates, and I successfully established the synthesis and autoassembly of azo-Tab DNA coacervates inside CM-dextran DA-dextran coacervates. Such multiphase organization is due to differences in surface tension and ionic strength between these two coacervates, where the coacervate with the highest ionic strength is usually present as an internal phase. Upon doping with TAMRA ssDNA, the fluorescent oligonucleotide is internalized in the light-responsive coacervate, and after shining UV light, we induce the disassembly and the release of the payload into the CM-dextran DA-dextran phase. With confocal microscopy, we could observe that the fluorescent oligonucleotide and C-fluorescein internalized in the two different coexisting phases. And upon UV irradiation, we could release the fluorescent oligonucleotide into the external phase. We were also successfully expanding this multiphase system to include azo-Tab ATP coacervates as an internal phase. And we could reform the internal phase upon shining blue light on the system. And here you could see the nucleation of the azo-Tab ATP phase. And this internal phase is forming and growing over time. Currently, we are looking at the inclusion of photoresponsive droplets into giant unilamellar vesicles with our German collaborators. We are inspired by their recent work where they assembled pH-responsive coacervates inside UVs that are based on POPC, cholesterol, DID, and formed via lipid film hydration. The last project that I'm presenting today concerns the shape modulation or vacuolarization of light-responsive coacervates. With blue light, we successfully formed vacuoles inside azo-Tab DNA coacervates. These vacuoles can form, grow, and decay in cycles, as shown in this video, acquired using confocal microscopy. And here you can see the cycle of growth, formation, and decay of these vacuoles. The idea was to stabilize the formation of these vacuoles via adding a third component that should not form photoresponsive droplets with DNA. We formed single-phase coacervates using azobenzene, polylysine, and DNA, and with UV light, we induced the formation of stable vacuoles inside these droplets. We can see that the size of the droplets did not change over time, but we can clearly see the nucleation and growth of these vacuoles and then their stabilization over time. We also looked at the fluorescent dye and oligonucleotides trafficking in these systems using confocal microscopy. And here we can see that the initially non-internalized fluorescein was able to internalize in these droplets upon forming the vacuoles and shining UV light on them. Whereas initially internalized TAMRA-read ssDNA is excluded from the droplets upon shining UV light on them and forming the vacuoles. To summarize, in my research work, I am relying on a bottom-up approach to assemble systems with lifelike properties. Of particular interest, light-responsive membraneless organelles can dynamically control the compartmentalization of polynucleotides, polypeptides, polymers, and proteins. Thank you for your attention.